First, thank you, Carmen, for inviting me to participate of this meeting. I am from Butantan Institute, Brazil, and good morning, everyone. And today I'm going to talk about the potential use of Brazilian marine macroalgae in schistosomiasis control. Well, schistosomiasis is a parasitic disease caused by worms of the genus Schistosoma, which affects more than 200 million people in the world. Brazil, although classified as a low transmission area, has more than 6 million people infected and 25 million exposed to the risk of infection. The only schistosoma species present in Brazil is Schistosoma mansoni. The disease is prevalent in 19 of the 27 states of Brazil. Well, regarding the intermediate host snails, we have in Brazil 3 species of Bionfalaria snails found naturally infected by Schistosoma mansoni. Bionfalaria glabrata, Bionfalaria stramínea, and Bionfalaria tenagophila. Well, currently, the chemical arsenal to the control of schistosomiasis is limited to only 2 drugs. Praziquantel is the only drug recommended for the treatment and control of schistosomiasis. And we have only 1 molluscicide, niclosamide, to the control of the intermediate host snails. So, searching for alternative drugs is highly recommended. But why the choice of seaweeds? Well, Brazil has more than 770 seaweed species distributed along a coastline of more than 7,300 kilometers at diverse ecological conditions. Well, the potential of seaweeds as a source of active compounds has already been demonstrated. Specifically on the anti-parasitic activity, there are several studies demonstrating the presence of active compounds on seaweeds. However, most of these studies come from protozoan, and there are only a few studies on the antiomimetic activity of seaweeds. And so far, there are no data on the activity of seaweeds on schistosoma worms. So, in order to select the seaweed species for our study, we've made a comprehensive screening with the 3 groups of macroalgae. The sampling was made in the coast of the Espírito Santo state. And in order to assess the potential of seaweeds as a source of active compounds against schistosomiasis, we've made a collection of 54 extracts from 41 seaweed species. Well, at this point, it's important to highlight that the molecular targets for schistosomicide and for molluscicide drugs are still unknown. So, we can't search for specific classes of compounds. So, in order to isolate the active compounds from seaweeds, we use traditional techniques. Lyophilized seaweeds are extracted with solvents of different polarities and tested for biological activities. Active extracts are fractioned through high-performance liquid chromatography, and the fractions are monitored through thin layer chromatography and nuclear magnetic resonance. And the active fractions are refractioned, and the process is repeated until the isolation of the active compounds. To elucidate the structure of the isolated compounds, we use spectroscopic techniques as infrared and mass spectrometry, nuclear magnetic resonance, and optical rotation. Well, we maintain the whole life cycle of Schistosoma mansoni in our laboratory in hamsters and Bionfalaria glabrata snails. Well, female hamsters are infected through injection of cercaria, and after 42 days, myrocidia are shed from the eggs taken from the liver. To the infection of snails, myrocidia are placed with the snails in culture plates for 3 hours, and after 45 days, snails are exposed to light to shed mature cercaria. To the in vitro test with Schistosoma mansoni, adult worms are obtained through portal perfusion. 5 pairs of worms are exposed to each concentration and assessed for 96 hours for alterations in motility, pairing, morphology, and reproduction. To the monocyte screening, we follow the guidelines of the World Health Organization, which establishes that crude extracts should be active at concentrations lower than 100 parts per million. Both adult snails and embryos can be used in this screening, but in our protocol we use embryos because of some advantages. The most important is to reduce the amount of material. So at least 50 embryos are exposed to test drugs and for 24 hours and assessed for 7 days for the induction of malformations and death. Well, regarding the results of this first trial, and considering only those species with 100% of activity, we had among the 26 species of red seaweeds, 6 active in Schistosoma mansoni and 14 active in Bionfalaria glabrata. Among the 11 species of brown seaweeds, we had 2 active in Schistosoma mansoni and 5 active in Bionfalaria glabrata. And among the 4 species of green seaweeds, we have 2 active in Bionfalaria glabrata. So, in total, 23 of the 41 seaweed species were active in at least one of the 2 models, which shows that Brazilian seaweeds can be a good source of active compounds against schistosomiasis. We have selected some species to isolate the active compounds. Octode secundiramia, Laurencia dendroidea, and some species of Dictyota are being submitted to bioguided fractioning. And also 16 compounds were isolated from Laurencia gingensis through nuclear magnetic resonance guided fractioning. So far, we have identified some active compounds, a monoterpene from Octode secundiramia, the isoptazole from Laurencia dendroidea, a dihydroceramide from Laurencia gingensis, and a sesquiterpene from Laurencia gingensis. Well, this work is only possible because we are a multidisciplinary team, and that's it. Thank you.